Welcome to Easy Microbiology. We're here to share our insights and experiences surrounding the topic of Enterococcus casseliflavus. We look forward to going through the various elements of our discussion, such as habitat, cultural characteristics, host range, prevention, treatment, and many more in the coming slides. Enterococcus casseliflavus is a type of bacteria that is gram-positive, meaning it retains a specific stain during laboratory testing. It is a facultative anaerobe, which means it can survive with or without oxygen. This bacterium is classified under the genus Enterococcus. Enterococcus casseliflavus is commonly found as a part of the normal bacteria population in the gastrointestinal tract. It can also be isolated from different clinical samples, including blood, urine, and wounds. This suggests that it has the potential to cause infections in these areas. Similarly, Enterococcus casseliflavus can be found not only in the human gastrointestinal tract but also in other environments such as soil, water, and various animals. This indicates that the bacterium has a wide distribution and can adapt to different habitats. In soil and water, Enterococcus casseliflavus may play a role in nutrient cycling and decomposition processes. It can also be present in animals, either as a part of their normal flora or as a potential source of infection. Understanding the diverse ecological niches where Enterococcus casseliflavus can be found is important for assessing its potential impact on human health and the environment. It highlights the need for proper hygiene practices and monitoring to prevent the spread of this bacterium and associated infections.
Enterococcus casseliflavus is a spherical bacteria that usually appears in pairs or chains when viewed under a microscope. It is a non-motile bacterium with a size ranging from 0.5 to 1.5 micrometers. This bacterium is commonly found in soil, water, and uncooked food products. Enterococcus casseliflavus is renowned for its ability to grow well on blood agar medium and its low susceptibility to aminoglycosides and cephalosporins. This bacteria is also characterized by its catalase negative status. All these cultural characteristics thus make Enterococcus casseliflavus a highly distinct species.
Enterococcus casseliflavus is characterized by its positive reaction for the enzyme pyrrolidinyl arylamidase, PYR. This enzymatic activity is useful for identifying and differentiating this bacterium from other Enterococcus species. Additionally, Enterococcus casseliflavus has the ability to ferment several sugars, including glucose, lactose, and mannitol. This means that it can utilize these sugars as a source of energy and produce byproducts such as acids or gases during the fermentation process. These biochemical characteristics are important for the identification and classification of Enterococcus casseliflavus in laboratory settings. They provide valuable information about its metabolic capabilities and help in distinguishing it from other related bacteria.
Enterococcus casseliflavus is a human pathogen with a wide host range that has been known to infect other animal species such as pigs, dogs, and chickens. This makes it an important factor to consider when assessing the risk of transmission from animals to humans.
Indeed, Enterococcus casseliflavus has the capability to produce various virulence factors that contribute to its pathogenic potential. One such factor is biofilm formation, where the bacterium can form a protective matrix of cells on surfaces, making it difficult to eradicate and increasing its resistance to antibiotics. Additionally, Enterococcus casseliflavus possesses the ability to adhere to host cells. This adherence allows the bacterium to establish and maintain colonization in various host tissues, which can lead to the development of infections. The production of these virulence factors by Enterococcus casseliflavus enhances its ability to cause disease and evade the immune system. Understanding these mechanisms is crucial for developing effective strategies to prevent and treat infections caused by this bacterium.
Enterococcus casseliflavus has been found to be inherently resistant to many antibiotics, and it is able to acquire resistance genes through horizontal gene transfer. This transfer of new genetic material between two organisms may bring about genetic traits and characteristics that may be beneficial for survival. Exposure to antibiotics can further increase this process, as it builds up the chances of horizontal gene transfer, thus leading to the spread of antibiotic resistance. It is imperative to have knowledge of this process and its results, in order to prevent the transmission of antibiotic resistance and to protect public health. Epidemiological research has shown that Enterococcus casseliflavus can cause infections in immunocompromised individuals, mostly in those with cancer or HIV. Its effects are particularly evident when the immune system of these patients is weakened, as Enterococcus casseliflavus is an opportunistic pathogen. The image shown here provides a close-up of the cells related to this pathogen.
Enterococcus casseliflavus has the potential to cause a range of infections in humans. This includes urinary tract infections, UTIs, where the bacterium can colonize and infect the urinary system, leading to symptoms such as frequent urination, pain, and discomfort. Bacteremia, another infection caused by Enterococcus casseliflavus, occurs when the bacterium enters the bloodstream. This can happen through wounds, invasive procedures, or from other sites of infection. Bacteremia can result in serious complications and may require prompt medical intervention. Wound infections are also associated with Enterococcus casseliflavus. When the bacterium enters a wound, it can multiply and cause localized infection, leading to delayed wound healing and potential complications. Recognizing the potential for Enterococcus casseliflavus to cause these infections is important for appropriate diagnosis and treatment. Prompt identification and targeted therapy are crucial in managing and preventing the spread of these infections.
The human body is known to produce antibodies and activate T cells in response to Enterococcus casseliflavus infections. This is its natural process of combating and defending against infection, which can vary from individual to individual, depending on the type of infection. In diagnosing Enterococcus casseliflavus, standard microbiological techniques such as blood culture and biochemical tests are used. These tests involve cultivating the organism under laboratory conditions and testing its reaction to various materials. This allows a microbiologist to accurately identify the presence of Enterococcus casseliflavus. Knowing the presence of this organism gives the healthcare team the knowledge to make an informed decision and plan the best course of care. Good hygiene is a key factor in preventing Enterococcus casseliflavus infections. This includes regular and thorough hand washing, as well as cleaning and disinfecting equipment, surfaces, and materials that may have come in contact with the organism. In addition, if appropriate, antibiotics can help inhibit the growth of Enterococcus casseliflavus. Appropriate use of antibiotics includes making sure the right drug is selected for the infection, following the instructions provided by a healthcare professional for the correct dosage and duration of treatment. Treatment of Enterococcus casseliflavus infection typically involves antibiotics. Penicillin and vancomycin are typically used for this type of infection. However, it is important to note that these infections may require a specific antibiotic and may not respond to generic antibiotics.
Enterococcus casseliflavus is a gram-positive bacterium that normally resides in the gastrointestinal tract of humans as a part of the normal flora. However, in individuals with weakened immune systems, it can cause infections. One concerning aspect of Enterococcus casseliflavus is its inherent resistance to many antibiotics. This means that it naturally possesses mechanisms that make it difficult to treat with commonly used antibiotics. Furthermore, Enterococcus casseliflavus has the ability to acquire resistance genes through a process called horizontal gene transfer. This means that it can exchange genetic material with other bacteria, potentially acquiring additional resistance mechanisms and further reducing treatment options. The combination of inherent antibiotic resistance and the potential for acquiring new resistance genes makes Enterococcus casseliflavus a challenging bacterium to manage and treat, particularly in immunocompromised individuals. Careful monitoring and appropriate antibiotic stewardship are necessary to prevent the spread and emergence of antibiotic-resistant strains. Thank you for watching. If you found this information helpful, please consider liking, sharing, and subscribing to my channel. Your support is greatly appreciated.